Good day, everyone, and welcome to another session in our lecture discussion for biochemistry. Now, for this discussion po, we will be covering the topic carbohydrate metabolism. Now, carbohydrate metabolism is a fundamental biochemical process that ensures a constant supply po of energy to living cells. Now, in this chapter, we will explore the relationship between carbohydrate metabolism and energy production in cells. Now, the molecule, now glucose, is the focal point of carbohydrate metabolism. Commonly called blood sugar, glucose is supplied to the body via the circulatory system, and after being absorbed by the cell, it can be either oxidized, no, to yield energy, or stored as glycogen, no, for future use. Now, as discussed during the previous discussion po of biochemical energy production, digestion is the first stage in the processing of food products. Now, it is the biochemical process by which food molecules, through hydrolysis, are broken down into smaller and simpler chemical units that can be used by cells for their metabolic needs. Now, the digestion of carbohydrates will start in the mouth once the food is swallowed. Now, the mouth contains the enzyme salivary alpha amylase that catalyzes the hydrolysis of alpha glycosidic linkages in starch, no, from plants and glycogen from meats that we ingest, now to produce smaller polysaccharides and a disaccharide maltose. Now, only a small amount of carbohydrate digestion happens in the mouth. So, because the food is swallowed quickly, and pag nasa stomach na po, no, also a little carbohydrate digestion occurs either since the salivary alpha amylase is inactivated by the acidic environment of the stomach, and the stomach do not contain any carbohydrate digesting enzymes. Now, the primary site for carbohydrate digestion is within the small intestine, where alpha amylase, this time, secretes by the pancreas, begins now to function. The pancreatic alpha amylase breaks down polysaccharide chains into shorter and shorter segments until the disaccharide maltose and glucose itself are the dominant species. As a review po, what is the component of maltose? So the answer po would be two glucose units. Now, the final step here in the carbohydrate digestion occurs in the outer membranes of intestinal mucosal cells, where the enzymes that convert disaccharides to monosaccharides are located. The very important disaccharides enzymes like maltase, sucrase, and lactase are located here. Now, maltase will convert maltose to two glucose units. Sucrase naman po, on the other hand, will convert sucrose to one glucose and one fructose unit. And lactase will convert lactose to one galactose and one glucose unit. Now, take note that the disaccharide sucrose and lactose present in food are not digested until they reach this point. Now, glucose, galactose, and fructose are the three major breakdown products from carbohydrate digestion. Now, these products are absorbed into the bloodstream through the intestinal wall. Now, as we all know, small intestine contains finger-like projections known as villi, which functions for absorption. Absorption po is via active transport, meaning it requires energy or ATP molecules for the process. Now, after the absorption of the end products of carbohydrate metabolism into the bloodstream, these monosaccharides po are transported to the liver, where fructose and galactose are rapidly converted into compounds that are metabolized by the same pathway as glucose. Thus, the central focus of carbohydrate metabolism is the pathway by which glucose is further processed, a pathway called glycolysis. Now, the glycolysis po is the conversion of glucose to pyruvic acid, which happens po in a process called oxidation, in which this oxidation po happens naman without the presence of oxygen. Now, the oxidizing agent is the coenzyme NAD. Thus, we can call glycolysis as a type of anaerobic pathway. Now, glycolysis po is a 10-step process, now, in which every step is enzyme catalyzed. Now, there are two stages in the overall process, a 6-carbon stage and a 3-carbon stage. Okay, before we discuss the stages in of glycolysis, now a brief history. Glycolysis po is also called as Embden-Meyerhof pathway after the German chemists Gustav Ebden and Otto Meyerhof discovered the many details of the pathway in the early 1930s. Now, let us discuss now the two stages of glycolysis. The first stage, the 6-carbon stage of glycolysis, is an energy consuming stage. So please take note. Not the this stage po is an energy consuming stage. So the intermediates of the 6 carbon stage of glycolysis are all either glucose or fructose derivatives in which phosphate groups are present. Now glycolysis begins with the phosphorylation of glucose to yield glucose 6 phosphate, which is the step 1. Glucose 6 phosphate po is a glucose molecule with a phosphate group attached to the hydroxyl oxygen on carbon 6. So the carbon atom outside the ring. Now the phosphate group is from an ATP molecule, and the major enzyme in the step 1 would be your hexokinase, which is an enzyme that requires magnesium ion for its activity to catalyze the reaction. Now, on the second step of the 6 carbon stage of glycolysis, glucose 6 phosphate now is isomerized to fructose 6 phosphate by the enzyme called phosphoglucoisomerase. Now, as you can see here, the net result of this change is that carbon 1 of glucose is no longer part of the ring structure. Now, like step 1 po, step 3 is a phosphorylation and therefore requires energy. Now, ATP is the source of the phosphate and the energy here in the step 3. Now, the enzyme involved here is your phosphofructokinase, which is another enzyme that requires magnesium for its activity. So, as you can see here, the fructose 6 phosphate is converted into fructose 1,6 bisphosphate by this enzyme. Now, the result po is a 2 phosphate group. Groups are attached po to a fructose molecule in the first and the sixth carbon of the structure. Okay, so we are now done on the steps 1, 2, 3, which are under the 6 carbon stage of glycolysis. Now, entering the 3 carbon stage of glycolysis, so the 3 carbon stage of glycolysis po naman is an energy generating stage rather than an energy consuming stage compared to the 6 carbon stage po. Now, all of the intermediates naman in this stage are carbon 3 phosphates or C3 phosphates, two of which are high energy phosphate species. Now, the C3 intermediates in this stage of glycolysis are all phosphorylated derivatives of dihydroxyacetone, glyceraldehyde, glycerate, or pyruvic acid, which in turn are derivatives of either glycerol or acetone. Now, in the fourth step, no, which involves cleavage, so the reacting C6PC from the previous step, which is your fructose 1,6 bisphosphate, is split into two triose species. So that's why this stage is called as 3 carbon stage. So, no, pag na-split na po, no, unsymmetrically, yung ating fructose 1,6 bisphosphate, it will yield a product, which is your dihydroxyacetone phosphate, and the other is glyceraldehyde 3 phosphate. Now, the two trios po produced during the step 4, which is your dihydroxyacetone phosphate, a type of ketose, and the glyceraldehyde 3 phosphate, which is a type of an aldose, are isomers of each other. Now, for your glyceraldehyde 3 phosphate, it can be directly be converted or it is directly oxidized po and phosphorylized in the step 6. However, your dihydroxyacetone phosphate it should be converted first into another glyceraldehyde phosphate via the process of isomerization for it to be accepted on the step 6 by the enzyme involved in the step 6. So this step po, the step 5, which involves isomerization of your dihydroxyacetone phosphate is also needed no to convert this dihydroxyacetone into glyceraldehyde 3 phosphate. Now starting from this step, no, magiging dalawa na po yung glyceraldehyde 3 phosphate na ipaprocess on the step 6. Now, on the step 6 naman po, it involves oxidation and phosphorylation of your glyceraldehyde 3 phosphate coming from the step 4 and 5 previously. Now, as you can see here, by the uh, reaction catalyzed by the enzyme glyceraldehyde 3 phosphate dehydrogenase, a phosphate group is added to the glyceraldehyde 3 phosphate to produce the product 1,3-bisphosphoglycerate. Now, as you can see here, the newly added phosphate group in the structure of your 1,3-bisphosphoglycerate is a type of a high energy phosphate group. As we discussed previously, no, a high energy phosphate group is produced when a phosphate group is attached to a carbon atom that is also participating in a carbon-carbon or carbon-oxygen double bond. So, also in the structure here, hindi lang masyado makita. So, a squiggle notation is added on the uh, writing of the structure of your 1,3-bis phosphoglycerate, indicating a high stress or high energy phosphate group. As you can see also here, byproducts of step 6 is an NADH, a reduced form of NAD, and also a hydrogen atom. Now, the next step will be your step 7, a type of phosphorylation of the structure. Now, in this step po, as you can see, the diphosphate species coming from the step 6 is converted back to a monophosphate species via the action of the enzyme phosphoglycerokinase. Now, this step naman po is a type of ATP producing step in which the C1 no, phosphate group of the 1,3-bis phosphoglycerate is transferred to an ADP, adenosine diphosphate molecule, to form the ATP. So to form the product 3-phosphoglycerate. So yung phosphate na kinabit ng step 6 doon sa pinaka-top portion ng structure, tatanggalin ulit ng enzyme na to. And ita-transfer po niya yung natanggal na phosphate in ADP to produce ATP now po. Okay? Now, this is called a substrate level phosphorylation, okay, in which a high energy phosphate group from an intermediate compound is directly transferred to ADP to produce ATP. So please take note, step 7 is an energy producing stage. So on the previous discussion, let us review. Step 1 and 3 is a type of energy consuming stage, while step 7 is the first step of an energy generating step. Now, step 8 po involves another isomerization where the phosphate group of 3-phosphoglycerate is moved from carbon 3 no, up into the carbon 2. Now, the enzyme phosphoglyceromutase catalyzes the exchange of the phosphate group between the two carbons. Now, after the carbon po is transferred from the C3 up until the C2, which produces 2-phosphoglycerate, it undergoes the dehydration process in step 9. Now, this is a type of an alcohol dehydration reaction that proceeds with the enzyme enolase, another type of enzyme that requires magnesium ion. Now, the result here is another compound containing a high energy phosphate group. Now, the phosphate group is attached to a carbon atom that is involved in a carbon-carbon double bond. Therefore, this is a type of energy, a high energy phosphate molecule again. Then, in the step 9 also, we can see the byproduct of alcohol here. Now, in the last step of glycolysis, another substrate level phosphorylation occurs. Now, phosphoenol pyruvate, the enzyme involved here, transfers its high energy phosphate group to an ADP again to produce ATP and pyruvic acid. So, ang pinaka-goal po ng step 10 natin is to produce pyruvic acid. Now, the enzyme involved, pyruvate kinase, requires both magnesium and potassium ions for its activity. Now again, because two C3 molecules are reacting, starting from step 4, okay, or 5, two ATP molecules are produced. In this table shows you po the number of products no, or number of ATP that is consumed and produced in all the steps of your glycolysis. Now take note that ATP molecules are involved in steps 1, 3, 7, and 10 of glycolysis. Now considering these steps collectively shows that there is a net gain of 2 ATP molecules for every glucose molecule converted into pyruvates. So please remember the steps 1 and 3 are energy consuming stage. So kung ano yung na-produced from your 7 and 10, which is a double product of ATP, dun lang din sila kukuha ng ATP to function. That's why the net ATP from all the steps of your glycolysis will produce 2 ATP po. Okay? So kung makikita natin, onti siya. Pero yung pyruvic acid kasi na ginawa ng ating glycolysis, syempre will undergo reprocessing and then it will be attached to a type of coenzyme A, and then that coenzyme A will be uh, will be transformed into acetyl coenzyme A that will be used naman to start up the Krebs cycle. Now, in this slide shows the summary of the 10-step process of glycolysis, which involves many types of enzymes, so including your hexokinase, your phosphoglucoisomerase, your PFK, your aldolases, your triose phosphate isomerase, your GAP DH, your phosphoglycerate kinase, and also your phosphoglyceromutase, phenolase, and your pyruvate kinase. Now, the breakdown products po from carbohydrate digestion again are glucose, fructose, and galactose. Now, both fructose and galactose po, please take note, are converted in the liver to intermediates that enter into the glycolysis pathway, as you can see in this uh, slide. Now, the entry of fructose into the glycolytic pathway involves phosphorylation now by ATP now to produce fructose 1 phosphate, which is then split into two trios, your glyceraldehyde and also your dihydroxyacetone phosphate. Now, your dihydroxyacetone phosphate enters glycolysis directly. A glyceraldehyde naman po must be phosphorylated by ATP first to your glyceraldehyde 3 phosphate before it enters the pathway. Pero syempre, your dihydroxyacetone phosphate is isomerized again to glyceraldehyde 3 phosphate. Now, the entry naman po of galactose into the glycolytic pathway begins its conversion to glucose 1 phosphate, a four-step sequence, which is not included here, which is then converted to glucose 6 phosphate, an intermediate po in between the step 1 and 2. Now, the production of your pyruvic acid or pyruvate from glucose occurs in a similar manner in most cells. In contrast, the fate of the pyruvate so produced varies with cellular conditions and the nature of the organism. Now, three common fates of pyruvate are of prime importance. First is the conversion into acetyl coenzyme A, which is mentioned earlier. It can also be converted into lactic acid or lactate, and it can be also converted into ethanol by some organisms. So take note, our body, no, our human cells can under aerobic conditions, it can convert your pyruvic acid into acetyl coenzyme A, also in animals and other microorganisms. In anaerobic conditions naman po, in also humans, animals, and other microorganisms, your pyruvic acid naman in anaerobic condition is converted into lactic acid. One classic example of your pyruvate converting into lactic acid is when your muscles po, no, work beyond its normal processing. So kapag ka anaerobic na yung process sa muscles, the pyruvic acid is converted into lactic acid. That's why your muscles after workout or overwork, no, tends to have a kind of pain. That pain po comes from the accumulation po of the lactic acid in the muscle tissues. Now, a key concept po in considering these rates of pyruvic acid is the need for a continuous supply of NAD for glycolysis. Now, as glucose is oxidized to pyruvic acid in glycolysis, NAD is reduced po to NADH. So, it is significant that each pathway of pyruvate metabolism includes provision for regeneration of your NAD from NADH so that your glycolysis can continue. Now again, under aerobic or oxygen-rich conditions, your pyruvate po is oxidized to your acetyl coenzyme A, as you can see here. Now in the cytosol or cytoplasm, the pyruvic acid is formed through glycolysis. Now the overall reaction in simplified terms is like this. So pyruvate na formed in the cytosol through glycolysis crosses the two mitochondrial membranes and enters the mitochondrial matrix where the oxidation takes place. Now, the major enzyme involved here is your pyruvate dehydrogenase complex. Now, the byproducts naman po of this oxidation of your pyruvic acid would be NADH and carbon dioxide. Then carbon dioxide is further exhaled, no, in the body via your lungs or your blood into your lungs. Other footnotes regarding the oxidation of your of your pyruvic into acetyl coenzyme A. So please take note that this reaction involves both oxidation and decarboxylation because it produces carbon dioxide. Now, the overall reaction process involves four separate steps and requires NAD, coenzyme ASH, and your FAD, and two other coenzymes, which are your lipoic acid and thiamine pyrophosphate, which is a derivative of B vitamin thiamine. Now, most acetyl coenzyme A molecules produced from pyruvate enter the citric acid cycle. Now, in cases naman po when your body becomes oxygen deficient or it undergoes anaerobic conditions such as during strenuous exercise, now the electron transport chain process slows down. So because because it is the last step po which is dependent on oxygen. Now, the result of this slowing down of your ETC or electron transport chain, no, build produces a build-up in NADH concentration. So meaning it is not being consumed so fast. And a decreased amount of available NAD is also produced or it is not being produced so fast naman. Now, decreased NAD concentration po negatively affects the rate of glycolysis. So, nagkakaroon ng negative feedback mechanism. An alternative method po for conversion of NADH to NAD, which does not require oxygen, is needed if glycolysis is to continue. Now, the fermentation process solves this problem now in certain types of cells or uso rin po ito lalo na sa mga microorganisms. Okay? So fermentation processes solves this problem, no? By converting NADH, no, or oxidizing NADH to NAD again without the need for oxygen. Okay? So we consider two fermentation process, which are your lactate fermentation and ethanol fermentation. Now, the enzymatic anaerobic reduction of pyruvate to lactate is known as lactate fermentation. Okay, as you can see here. So pyruvate needing NADH, no, by the enzyme lactate dehydrogenase. Meron po tayo nitong enzyme na to, will convert your pyruvate to lactate, no, producing oxidized form, no, of NADH, which is your NAD. So again, note that NADH and NAD do not appear in this equation, so as in this lactate fermentation, even though the process cannot proceed without them. Now, the NADH generated during glycolysis or the step 6 of glycolysis is consumed in the conversion of pyruvate to lactate. Thus, there is no net oxidation reduction in the conversion of glucose to lactate. Now, under anaerobic conditions, again, several simple organisms naman, including your yeast, possess the ability po to regenerate NAD through ethanol rather than your lactic acid kanina. Okay? So, such a process is called ethanol fermentation. Now, ethanol fermentation po involving yeast causes bread and related products to rise as a result of the production of carbon dioxide bubbles being released during baking. Now, beer, wine, and other alcoholic drinks are produced by ethanol fermentation of the sugars in grain and fruit products. Now, ethanol fermentation po is the enzymatic anaerobic conversion of pyruvic acid to ethanol and carbon dioxide. Now, the first step in conversion of pyruvate to ethanol is a decarboxylation reaction to produce acetaldehyde, as you can see here, by the enzyme called pyruvate decarboxylase, releasing po additionally carbon dioxide in the process. Now, the second step naman po of ethanol fermentation involves acetaldehyde reduction to produce now your ethanol by the enzyme called alcohol dehydrogenase. So again, note that NADH and NAD do not appear in the final equation. So they are both generated and consumed. So an overall reaction for the production of ethanol from glucose is obtained by combining the reaction for the conversion of pyruvate with a net reaction for glycolysis. Now, this figure naman po summarizes the relationship between the fates of pyruvate and the regeneration of NAD from NADH. Now, let us summarize for the ATP production for the complete oxidation of glucose. Now, the new piece of information po involves the NADH produced during step 6 of glycolysis. Now, this NADH po na napoproduce on the step 6 of glycolysis, produced primarily in the cytosol, cannot directly participate in the electron transport chain. So, because mitochondria are impermeable to NADH. So, small molecules lang po are allowed to enter the outer membrane of your mitochondria. Now, because of this one, so a specific transport system shuttles the electron from NADH. Now, but not NADH itself, no? So this shuttle involves the hydroxyacetone phosphate, the intermediate of glycolysis, and glycerol 3 phosphate. Okay? So, the hydroxyacetone phosphate and glycerol phosphate freely cross kasi po the mitochondrial membrane. Now, the interconversion shuttles the electrons from NADH, no, down to your FADH2. So, hindi talaga pumapasok per se or dinadala per se nung dihydroxyacetone phosphate and glycerol phosphate yung malaking molecule na NADH. So, kinukuha lang nila yung electrons and then they will transfer it to your FADH2. Okay, we are now finished discussing the step-by-step process of your glycolysis. Now, in our body and in animals also in other living organisms, if your carbohydrates no or if your glucose are in excess in the body, it is bad. Okay, so that's why in certain tissues like in muscle and liver tissues, your the storage form of glucose, which is your glycogen, is produced. So, it is in the process called glycogenesis. Now, glycogenesis po is the process of storing excess glucose for use by the body at a later time. It is the conversion of glucose into glycogen or its storage form. Now, glycogenesis involves three reactions. Now, the process starts with a substance called glucose 6 phosphate, which, if you remember, is an intermediate product in the step one no of the glycolysis, so which is available from the first step nga po. So your enzyme phosphoglucomutase changes the 6 phosphate into 1 phosphate of the structure. So the step 1 involves the formation of glucose 1 phosphate. Now, the second step of your glycogenesis involves the formation of your UDP glucose. Now, UDP or uridine diphosphate no, is involved here. Now, saan po manggagaling yung phosphate here? It comes from your uridine triphosphate. Now, uridine triphosphate or UTP, no, must activate po kasi your glucose 1 phosphate coming from the step 1. Okay? Before it can be added to a growing glycogen chain. Okay? So AUMP or uridine monophosphate is transferred to glucose 1 phosphate, and the resulting phosphate is hydrolyzed into two phosphate ions. Okay? So the activator here is the high energy compound UTP. Okay? So once activated na po, it will go na on the step 3. Now, the glucose unit of your UDP glucose is now attached no, to the end of your glycogen chain. So in the step 3 po, it involves the glucose transfer into a glycogen chain. So the glucose unit of UDP glucose is then attached here. So pagkakabit-kabit nilang po yan ng glycogen synthase hanggang makabuo po tayo ng glycogen molecule. No, and then in the byproduct po is the release no of your UDP. Then UDP is again used no or utilized by the cell no to produce other glycogen in this process. Now, in contrast po with the process called glycogenesis, your glycogenolysis naman po is the opposite of that process. Now, glycogenolysis, a process by which glycogen, the primary carbohydrate stored in the liver and muscle cells, is broken down into glucose naman po to provide immediate energy and to maintain blood glucose levels during fasting state. Now, your glycogenolysis involves two-step process. So the first step po involves the phosphorylation of a glucose residue. Now, this process is not simply the reverse of glycogen synthesis or glycogenesis because it does not require na UTP or UDP molecules. Now, glucose 1 phosphate from a glycogen molecule are removed no, during the first step via the action of the enzyme called glycogen phosphorylase. Now, the phosphate group naman po of glucose 1 phosphate is moved to the carbon 6 position via the process called isomerization with the help of this enzyme called phosphoglucometase. Now, this process is the reverse of the first step of glycogenesis. Now, starting from here, now glucose 6 phosphate now will enter now the step 2 of glycolysis to produce now ATP. Now, the stimulus po that initiates the glycogenolysis is the immediate need for energy in muscle and brain cells. Now, the glucose 6 phosphate that is produced directly enters the glycolysis pathway again at step 1, and its multi-step conversion to pyruvate begins. Now, while the stimulus that initiates glycogenolysis in liver cells is the low level of glucose. Now, the glucose 6 phosphate produced must be converted to free glucose before it can enter again in the bloodstream. So as glucose 6 phosphate. Now this change is affected by the enzyme glucose 6 phosphatase, which is an enzyme found in liver cells but not in muscle cells or brain cells. Now because muscle and brain cells lacks glucose 6 phosphatase, they cannot form free glucose from glucose 6 phosphate. Thus, your muscle and brain cells can use glucose 6 phosphate from glycogen for energy production only. Now, the liver, however, with this enzyme present, has the capacity to use glucose 6 phosphate obtained from glycogen to supply additional glucose to the blood. Now, another process besides the previously mentioned pathways, no, gluconeogenesis naman is another um, alternative pathway, no, to synthesize new glucose from non-carbohydrate precursors. So it provides glucose when dietary intake is insufficient or absent. So the non-carbohydrate starting materials for gluconeogenesis are lactate. Okay, so from hardworking muscles and from red blood cells, it can also be glycerol, no, from the triacyl glycerol hydrolysis or your tags. Okay, and certain amino acids from the dietary protein hydrolysis or from muscle protein during starvation. Now, about 90% of gluconeogenesis takes place in the liver. Hence, gluconeogenesis helps to maintain normal blood glucose levels in times of inadequate dietary carbohydrate intake such as between meals. Okay? So, ito yung nagme-maintain, okay, ng normal blood glucose levels natin pag di tayo kumakain. Okay, as you can see here in the representation, so the pathway of glycolysis is similar with gluconeogenesis, but not identical. So the most obvious difference between these two processes is that 12 compounds now are involved in gluconeogenesis and only 11 in glycolysis. So magkabaliktad but still there is a difference po. So ano po yung difference sila? So sa gluconeogenesis, your pyruvate before it is converted to oxaloacetate before it is converted to phosphoenol pyruvate. Now, gluconeogenesis po using lactic acid as a source of pyruvic acid is particularly important because of lactate formation during strenuous exercise. Now, the lactate produced diffuses from muscle cells into the blood, or it is transported to the liver. Now, the enzyme lactate dehydrogenase, the same enzyme that catalyzes lactate formation in the muscle, converts your lactic acid back to pyruvate. So kapagka masakit yung muscle cells yung muscle natin, 'di ba? 'Pag nagpahinga lang tayo after strenuous exercise, nawawala siya. That is because it undergoes the Cori cycle. Okay? So the newly formed pyruvate is then converted via gluconeogenesis to glucose, which enters again the bloodstream and goes to the muscles. So this cyclic process again is called Cori cycle, which it is governed by your lactate dehydrogenase. So in the production of pyruvate from this process, the byproducts of this process is your NADH and your hydrogen ions. Now, this figure illustrates po the relationship of the four common metabolic pathways that involves glucose. So, glycogenesis, a three-step process involving glucose 6-phosphate units are added to a growing glycogen molecule. Now, gluconeogenesis naman po is a 11-step process that converts pyruvate into glucose. Then, glycolysis po again. So, to summarize, this is the first step of the glycolysis po is the production of glucose 6-phosphate from glucose. Then glucose 6-phosphate produced can be further processed in the glycolysis pathway or processed to glycogen using like glycogenesis pathway. Then glycogenesis naman po is the second step of glycogenesis. The second step here is the conversion of glucose 1-phosphate to UDP glucose, a type of activated form of the structure. Now, alternatively po, glycolysis is not the only pathway, no, by which glucose may be degraded. Okay? Now, depending on the type of cell, various amounts of glucose po are degraded now by the pentose phosphate pathway. So, a pathway whose main focus is not subsequent ATP production, just like for glycolysis. Now, the major functions of this alternative pathway are first, to synthesize the coenzyme NADPH needed in the lipid biosynthesis. Second is to produce ribose 5-phosphate, which is a type of a pentose derivative needed for the synthesis of nucleic acids naman po and many coenzymes. Now, there are two stages within the pentose phosphate pathway, the oxidative stage and a non-oxidative stage. Okay? So the oxidative stage po, which occurs first, involves three steps, no, through which your glucose 6-phosphate from your glycolysis is converted to ribulose 5-phosphate and carbon dioxide, as you can see here. Okay? Now, the second stage is the non-oxidative stage of the pentose phosphate pathway, which your ribulose 5-phosphate naman po is isomerized to ribose 5-phosphate, uh, which is ultimately converted into glyceraldehyde 3-phosphate and fructose 6-phosphate po. Okay? Now, in this table po, shows you the step-by-step process of your pentose phosphate pathway. So, marami pong branches ang pentose phosphate pathway. So, pwede po siyang patigilin at the very half of the pathway. So, that is the conversion of glucose 6-phosphate into ribulose 5-phosphate when needed. So, when needed po, pwede po siyang ituloy up until the last steps na to produce fructose 6-phosphate po in the procedure. Now, there are two main classes of enzymes involved in the pentose phosphate pathway, which are your transketolases and transaldolases. Now, in definition, transketolases are a key enzyme po in the non-oxidative branch of the pentose phosphate pathway, which transfers a 2-carbon aldehyde unit from the ketose donor to the aldose acceptor sugars. While your transaldolases naman is a key enzyme again in the non-oxidative branch naman po, which transfers naman 3-carbon aldehyde from your ketose donor naman to the aldose acceptor sugars. So as previously mentioned po, your pentose phosphate pathway po, pwede po siyang ituloy sa dulo or kalahati lang. So with its many intermediates, your pentose phosphate pathway kasi po, it helps meet the cellular needs in numerous ways. So for example, so if the cells needs a high demand for ATP, now the pathway continues up until the end products. So the end products of intermediates of your glycolysis that will enter the process. So when NADPH naman po is high, so or the demand of NADPH is high in the cell, the intermediates are recycled, no, to glucose 6-phosphate and further NADPH is produced. So hanggang dun lang po siya sa may uh, uh, up until the do'n lang po sa kalahati 'no, nung mismong process. Para paulit-ulitin po 'yun, pabalik-balik siya dun sa umpisa, hanggang sa maka-produce ng enough NADPH for the cell. So when ribose 5-phosphate naman, demand is high, which, in the case of, nucleic acid and coenzyme production, most of the non-oxidative stage is non-functional. So leaving ribose 5-phosphate as the major product. So up until lang siya doon sa may conversion ng ribulose 5 phosphate into ribose 5-phosphate. Now, this ribose 5-phosphate naman po is the starting material po of your nucleic acids and also coenzymes. Now, let's now discuss how we can control the carbohydrate metabolism. So, typically, no, your cells po follows the feedback mechanism in order for us to control, no? So, kung makikita natin kanina, so kung sobra yung glucose po in the body, it is stored po via glycogenesis. No? Kung kailangan naman po ulit, so pag na-sense naman po na onti lang yung substrate in the system, papataasin ulit yung glucose in the bloodstream by lysing the glycogen stored in the liver. So that is called the feedback mechanism, positive or negative feedback. Pero a second major method naman po to regulate carbohydrate metabolism, besides enzyme inhibition by metabolites, is hormonal control. Now, among others, three hormones like your insulin, glucagon, and epinephrine affect carbohydrate metabolism. So the release of insulin by the beta cells of the islets of Langerhans in the pancreas is triggered by your high blood glucose levels. Now, it lowers blood glucose levels by facilitating the entry, okay, of glucose into the cell. Therefore, it is a hypoglycemic hormone. Okay? So the glucagon naman po on the other hand is produced by the alpha cells of the islets of Langerhans in the pancreas, which is a hyperglycemic hormone naman. Okay? So it is released when blood glucose levels are low. Okay? So it functions to increase the blood glucose concentration by speeding up the conversion of glycogen to glucose, no, via glycogenolysis and gluconeogenesis respectively in the liver. Okay? So glucagon's effect is the opposite of insulin. Okay? Okay, the last hormone naman po on the hormonal control would be your epinephrine. Okay? So epinephrine naman or also called as adrenaline. So it is released by the adrenal glands in response to anger, fear, or excitement. Okay? So it functions similar to that of glucagon. So it stimulates glycogenolysis, which is the release nga po of glucose from your glycogen. So its primary target po are your muscle cells where energy po is needed for quick action. Okay? So that ends our discussion for carbohydrate metabolism. Thank you everyone for listening and have a good day.